Welcome everyone, my name is Till. I'm a Master of Science in Biotechnology, but today I would like to tell you a short story about bones and arteries. During the recruitment process for my position in the Intricare consortium, I had a Skype interview with my two main supervisors at Karolinska University, Assistant Professor Ljubica Matic and Professor Ulf Hedin, both outstanding researchers in the field of vascular biology, and in case of Ulf, also an excellent vascular surgeon. I was just coming off my master project in bone regeneration after fracture, so they were so nice to give me an overview and introduction into the field of atherosclerosis. Apparently, the vast majority of humans develop some form of fatty depositions within our arteries, which clinicians initially call fatty streaks. This occurs mostly due to aging and the constant turbulent forces that our vessel walls have to withstand in certain areas of the human body, much like a fast-flowing river would tear at the bank of a tight corner. High cholesterol, calcium and phosphate levels can lead to the deposition of more fat and inorganic crystals within the vessel wall. These foreign bodies will interfere with the natural structure of the vessel, and the immune system has to react to try and clear the debris. In order to avoid a rupture of the now weakened section, smooth muscle cells from the surrounding tissue will infiltrate and cover the now growing plaque and separate it from the lumen. In our modern society, high intake of fat and salt is not really a rare event, but it's much rather common practice. And that leads to the formation of very big, chronically inflamed plaques, which are the main cause for heart attacks and strokes, and therefore the biggest killer worldwide, leaving cancer in their dust. Nevertheless, our body possesses remarkable repair mechanisms and manages to keep the vessel function active for a very long time. So usually we don't even feel this constant struggle. We are asymptomatic, that's how they call it in the clinics. Clinical data derived from a human biobank, which my supervisors, together with collaborators at Karolinska University, formed and called BIKE, showed that high calcification in atherosclerotic patients correlated with asymptomatic patients and with symptomatic patients receiving the very established treatment with statins. Following this lead, they separated two cohorts of 20 patients each using computed tomography to rate them into high calcified versus low calcified. What they found is that high calcification correlated with an increase of smooth muscle function and an increase of extracellular matrix stability. Furthermore, it connected to lower inflammation and lower lipid content. In short, it seemed that high calcified plaques in late-stage atherosclerosis were more stable. Additionally, they found one target what was without a doubt the most highly upregulated gene in high calcification, namely PLG4, or commonly called lupersin. Lupersin is a big molecule which combines approximately equal parts of protein and sugar. So I call it the sugar chains red here, and they are the most important thing that leads to the function of lupersin, what it is mainly known for. They attract a lot of water and thereby enabling the lubricating function it has in our joints and tendons. Now, it also has a function in downregulating overshooting inflammation, which causes, for example, arthritis. So it was a very interesting target to study in connection with atherosclerosis. But at this point they stopped and they asked me, so where do you see yourself fit into this project? Do you think your previous experience could somehow help us? But yeah, I had some ideas. So all these calcifications, smooth muscle cells and inflammation reminded me of something that happens in open fractures. So when your bones protrude from the wound and the bone lining cells, which usually would initiate the healing process, are stripped away. In those cases, cells from the surrounding muscle tissue can infiltrate and express bone-like mechanisms which initiate and help close the gap between the bones and initiate the healing process. Well, of course, there's other clever people out there. I wasn't the first one to think like that. But in the field of atherosclerosis, there was like a battle going on already, with people having the same hypothesis, saying, okay, calcification is an active bone-like process. And people who say, no, that's not right. Calcification is a passive process and the deposition of mineralizing crystals. Now, I'm a pacifist, and I read a lot of good literature that was supporting both ways. So I was like, okay, why couldn't it just be like a combination of the two of them? So we formed the hypothesis that macrocalcification is a complex mechanism that leads to plaque stabilization finally and clinical risk reduction in late-stage atherosclerosis, with smooth muscle cells playing a major role exhibiting bone-like mechanisms. So the initiation of calcification in atherosclerosis is usually due to high levels of calcium and phosphate. So we wanted to see if these conditions would induce smooth muscle cells derived from patients that came to our clinics to upregulate genes that are usually found in cartilage and bone tissue. And indeed, we could show that these smooth muscle cells, when induced with high levels of calcium and phosphate, upregulated lubricin together with SOX9, which is a major driver of cartilage development. Also, we could see that already after 5 days, small calcifications shown here in red were forming around those cells. So there was clearly a calcification process going on. When we added additional lubricin to the culture and kept the overall same conditions, like from the picture it looked like it even increased this process. But when we looked at the genetic data again, we saw that lubricin, SOX9, and even genes that are usually upregulated in bone formation rather show the trend to be downregulated, which could only mean that the additional calcification was due to a passive process induced via the newly formed extracellular matrix, and that high levels of calcium and phosphate were inducing a phenotypic switch in smooth muscle cells being or acting like chondrocytes, but that lubricin itself didn't promote this process further. Next, we wanted to see at what stages and in what areas would lubricin be expressed during the disease. We looked at different models for vessel injury and plaque formation and plaque calcification, and even looked at pathological samples from humans. And what we found was that lubricin was an early reaction together with chondrocyte-like markers to vessel injury, and it preceded the formation of calcified plaques in mice and in humans. So to summarize a bit what we've learned so far is that high levels of calcium and phosphate can passively form small calcifications, which in turn induce cartilage-like gene expression levels in smooth muscle cells, which lead to the expression of lubricin, which is then trafficked out of the cell and forms a new extracellular matrix, which in turn interacts and precedes macrocalcifications. Finally, our clinical data could show that high calcification can be a stabilizing factor in late-stage atherosclerosis, which is in fact the usual state patients are in when they come to the clinics. So this is much like a concrete canalization of a river in a densely populated area. But it is important to somehow figure out the remodeling processes, because those are the dangerous times. And I believe advanced computed tomography, together with the detection of lubricin, could allow us to assess the grade of remodeling ongoing and act a little bit like a warning tape around the construction site to tell us, okay, here's something important going on. Thank you very much.